that leverage the power of epigenomics and 5-hydroxymethyl-cytosine, a novel DNA biomarker that is transforming how we detect cancer at its earliest stage. With more than 20 years of leadership across industry and academia, Sam has helped shape the field of molecular diagnostics from his early work at Celera Genomic Health to co-founding Lexant. Is it Lexant Bio, now part of Foundation Medicine? He has consistently been at the forefront of innovation. He is served on the faculty at the J. Craig Venter Institute, Scripps Translational Science Institute, and the Scripps Research. Today, we are proud to share that Human Longevity is launching Dr. Levy's technology, the Avantect pancreatic cancer test, as part of our advanced early detection platform. This marks a major milestone in our mission to eliminate preventable cancer death through proactive, data-driven health strategies. Dr. Levy's work is a powerful example of how science can save lives by detecting cancer before symptoms appear and giving patients a chance at the real prevention. Please join me in welcoming Dr. Sam Levy. Sam?
Thank you. Thank you very much for that very kind introduction, and a pleasure to be here. Thank you for the invitation to tell you about ClearNote Health's technology and specifically about some of the areas that we've been working in for early detection of pancreatic cancer. I think all too often we know that, alas, cancer is a leading cause of death worldwide, and typically this can be avoidable. This really can be avoidable. And because of the late diagnosis of disease, treatment options end up being very limited. So as you can see, this very dire statistic of 1 in 6 people are estimated to die from cancer. We really believe at ClearNote Health we can change that. And so some of the technology I'll tell you about today is really founded out of Steve Quake's lab at Stanford. So Steve has had a long background in genomics and technology development and single-cell genomics now. He's a visionary in the area of how technologies can really leverage changes in how we basically execute a range of different areas, diagnostics being one of them. And one of the things that he's talked about very early on when about 8 years ago, he and I were talking about technologies he's developed. He said that, really you need to focus in with cancer on parts of the genome that are turned on, genes that are actually moving in their dynamic modulation. And one of the ways in which you can do that is by measuring what I will show you next, this particular signal called hydroxymethylation. It's a change on the DNA. And if you can measure that with accuracy, Steve believed at the time, 8 years ago, that you can really develop tests that will actually detect cancer earlier. And I'm about to tell you that his vision proved to be the case. We've developed a range of different tests, and I'll describe one to you in detail. But if you look across the continuum of patient care in cancer, ranging from at the bottom of this slide, early detection, early diagnosis to treatment, then monitoring, then recurrence, what you'd really like to be able to do is have tests that enable you to detect cancers earlier. If you start this continuum of care with early detection, you facilitate the process by which a patient will do well in terms of their overall survival. And that's what we all obviously want to see. Avantect is a testing family focused on early cancer detection. And the technology, that 5-HMC signal that I told you about, is essentially what we have developed into 3 tests so far, 3 tests and counting, in the area of early detection for pancreatic, individually, ovarian cancer individually, and we have a test called for multi-cancer, which essentially captures all the cancers you see depicted in the center of this slide. The big right-hand side of this slide that's empty is not empty. We actually have a whole slew of technologies to look at therapy response. I will not focus on that today. I'll focus on primarily pancreatic cancer and how its detection is so crucial to happen early. What you'll be able to see is that if you can take the 64,000 patients diagnosed in 2023 for pancreatic cancer, alas, 50,000 of them will die from that disease. And for the most part, it's because of late detection and because patients are not symptomatic. And usually when they become symptomatic, they're too advanced in their disease. And pancreatic cancer is projected to be the second leading cause for cancer-related mortality in the US in these next 10 years. So we wanted to really see a change. This just puts into pictures more what I just told you, and that is that only about 11% of individuals have localized disease. That means it's restricted to the pancreas. And there's a potential, if you detect that early enough, you can do something to improve their survival. In fact, when you have localized disease, there's a 43% chance of having a 5-year relative survival. Contrast that now to individuals who most of the time have pancreatic cancer when it's diagnosed. They have distant disease. So the vast majority of patients, and those individuals with distant disease, have only a 3% chance of survival. So you can see early detection would increase your chances of survival by tenfold. And that's a very crucial statistic. And therefore, we want to move that detection curve to the earliest stages. What I'm about to tell you is about this pathway that you see depicted on the screen. You don't have to really understand this pathway beyond the fact that it's the change of the C letter in DNA, the cytosine base in DNA, when it goes through this cycle of methylation and then demethylation. And so this cycle in normal cells, non-cancer cells, continues. In cancer cells, it gets derailed. And the ability to detect a dynamic signal that signals active biology is this particular group that's added to the cytosine called hydroxymethyl. And this hydroxymethyl group is really interesting because a lot of biology has pointed to the fact it marks genes that are about to be turned on or turned off and how those genes are regulated. So it's a very crucial piece of the ability to detect cancer. In fact, when we looked on the left side of this slide, tumor tissues, and now you can compare the DNA from tumor tissues from pancreatic tumors to normal pancreas, you can see that in early stage versus late stage disease, there's a whole set of regions that have this hydroxymethylation signal. That's what each of these dots represents. And there's a very strong correlation between early stage and late stage that in pancreatic cancer is retained. Now, this is really important because what this means is you have the potential of detecting early stage disease. It's not just a problem where the patient will be detected in its latest stage. And in fact, that change in hydroxymethylation is also retained when you look at the cell-free DNA from the patient. So this is the DNA floating around in your blood. And that's really intriguing because that means that you can actually identify an individual, not have to do any kind of intervention, just draw their blood and answer the question, do they have pancreatic cancer? So I'm giving you a kind of foretaste of how we do this. And so this is how we do this. To start with, I showed you lots of dots on that previous slide. Which of those dots are really important? Each one of those dots was a gene that has a hydroxymethylation change. So which ones are most crucial? Well, this is how you figure it out. This slide depicts a way in which we use machine learning, AI, to actually discover which 5-HMC changes in which regions of the genome are important. So what we do is we take a set of patients with pancreatic cancer and individuals without cancer, and from their blood and ultimately plasma extract DNA, and then we run that DNA through these two assays. One of these assays is a whole genome sequencing assay, which gives you changes in the genome, copy number changes. These are insertions and deletions on genes that happen in cancer, and also changes in the DNA fragment size. Both of these events are very crucial in when tumors start manifesting in the body and how you can detect them in the plasma. And then in parallel, we run an assay which enriches the hydroxymethylation component of that DNA. And that enrichment, coupled with the whole genome sequencing, ends up generating a whole set of features across the genome. And with machine learning, we're able to take all these features and put them together in the classification algorithm. And that classification algorithm essentially emits two states. When you give it a patient sample, once it's trained on these 660 patients, it will tell you any new sample whether that patient has pancreatic cancer or not. And that's essentially how we train the algorithm. In fact, when you look at the algorithm, you see that of all the features in the algorithm, 80% of them are comprised of this hydroxymethylation event in the genome. Again, it comes back to Steve's original vision, which is look at the cancer signal, don't look at all the stuff around it. And so when you're able to measure this hydroxymethylation event, you can actually take that algorithm I just showed you on the previous slide, and you can ask the question, how well does it detect cancer in a completely different group of patients? And that's what we've done on the right-hand side. And my colleague David Hahn and others published it 2 years ago now. And with this, we took 2,150 patients. 2,000 of them didn't have cancer and about 100 did. And then we basically measured the performance of the algorithm. And what you have here is a measure of the performance. So specificity is a measure of how you accurately detect the negative group, the non-cancer group. And so 96.9% of the time you detect it accurately. And specificity is the flip side of our false positive rate. So 100% minus 96 is essentially our false positive rate, which is about 3%. And then sensitivity is how accurately you detect the actual cancer group. In this case, we're trending for early stage detection at 70%. And so with this, we compared how good is that? You're asking the reasonable question, is that good enough? Well, the only way that we have right now clinicians use, it's not recommended, but it's used, and that is for early detection. That's the glycan biomarker CA19-9. And we compared the performance of CA19-9 in this data set with the Avantect test. And you can see that for early stage, our ability to detect early stage cancer, stage 1 and stage 2, when you can really have surgery to help you recover, you can see that our performance and sensitivity is about 30% relatively higher for early stage detection with Avantect over CA19-9. And then similarly, in terms of performance, our Avantect test has a twofold lower false positive rate. So better in detecting cancer and better not giving a false alarm. And so both of those together made us feel that this is a really valuable test for patients. In fact, what I'm showing you here is our trajectory of the early evidence on the top left here for pancreatic cancer detection using 5-HMC. And this is all published in the peer-reviewed medical literature, to the validation study I just showed you, to a study that our lab director, Shimul Chowdhury, released that shows the validation of our test in our ClearCap lab. And then on the right-hand side, you see a poster from one of my colleagues, Anna Berger-Musky, that shows how well we detect across different modalities and risks of individuals with cancer. And I'll show you that next. But this gives us a lot of confidence that what we're aiming for has great value. We've, as you can see, released this as an early cancer detection test specific for pancreatic cancer. The early days of our work led us to submit the application of this test in a cohort of patients that I'll show you momentarily that have a high risk for disease. Those are individuals with new onset type 2 diabetes. And we got an FDA breakthrough designation for that technology, that it's novel breakthrough technology. And then in parallel, you can see that as we've shown how stable the test works in our lab and gone through all the regulation that's needed to get approvals and accreditations, Clear Cap, and then New York State, all of which took multiple years of time and effort with our team. I'm just very proud to say that we're in this place where we can offer this to patients through their clinicians. And in fact, for many clinicians, because of the performance, because of what the test is able to do, they've pulled the test into their clinical trials. And these clinical trials really inform how to use the test. So on the right-hand side, you can see there are 4 trials there. I'll tell you about 3 of them. The SAFE-D study in the UK, working with the National Health Service there, and that's essentially focused on new onset type 2 diabetes. The PROCEED study, which is looking at individuals with family history and genetic predisposition, and then the Kaiser Permanente study working with that group to look at a high-risk cohort using an electronic health record model. Very intriguing. I'll show you little pieces of data from each of these. The reason why it's so easy to use the test is because the test has a reporting that you see here. And this is a test report before I show you the studies. This test report is basically when the test is run in our Clear Cap New York State accredited lab. If there is an abnormal signal detected, this is the kind of report that is produced. In the small print, it gives you a little bit more information about what that signal means, how it comes about, and then what the next steps are. Consultation with a physician or licensed genetic professionals recommended. Additional clinical and radiological evaluation is required to establish a diagnosis, and hence the follow-on steps, right? This test is just the beginning of a trajectory to really help a patient address their cancer earlier. So next, I'm going to tell you about those studies I just mentioned, because they actually give you an entree into how we can use a test like this for a deadly disease like pancreatic cancer. So if you look across pancreatic cancer in clinical databases, you will note that about 10% of individuals have inherited disease. So they have 2 or more first-degree relatives, so close relatives that have had pancreatic cancer, and in some cases, a combination of genetic predisposition, germline mutations in their genome. And so knowing this is very valuable. 90% of individuals with pancreatic cancer have non-inherited or what's called sporadic disease. And so then the question is, how do you find those individuals? Well, there is a fact that individuals with new onset type 2 diabetes also have a higher risk for pancreatic cancer, and I'll show you that momentarily. And another aspect of pancreatic organ development is sometimes you have cysts, they're called IPMNs, in the pancreas, and these also put patients at higher risk. Now, cysts are quite common in the population, so not everybody with cysts has pancreatic cancer, obviously, but in some cases, tracking a cyst once seen is a very valuable and important aspect of risk management and surveillance. And so one thing that we focused on very early was compared to, on the left-hand side of this graph, where most people above the age of 50 years have an average risk for disease over a 3-year period. Individuals with new onset type 2 diabetes or family history have a 6 to 8-fold higher risk. And in fact, individuals with family history under guidelines are suggested to have routine surveillance as part of their care. So this is the area that we focused on initially. And it's quite a detailed slide, but just suffice to say, the study looking at new onset type 2 diabetes is underway in the UK. We're looking at 15,000 individuals, and our intent here is to randomize them to 2 study arms, one that gets Avantect, and depending on the test results, if it's positive, we'll go to MRI. If it's negative, we'll go through their standard care, and that happens at 3 time points across 2 years, and then they follow up at the end of the 3rd year. Now, what this is intended to show is what's the testing modality that will best capture and improve the ability to find pancreatic cancer earlier. And that's what this study arm shows. And the control arm is essentially what patients get as standard of care, which unfortunately for new onset type 2 diabetes is just a follow-up in case when a patient is symptomatic. And so this study will read out in another year or 2, at least partially, and it's going to tell us a lot more about the periodicity of testing. Another study is with PROCEED Consortium. PROCEED Consortium is at 50 sites across the US and another set of sites across internationally. And PROCEED is looking at these individuals with familial pancreatic cancer, or and additionally genetic predisposition risk. And so we're following these patients every year with our Avantect test. And in fact, you can see this pictorial just shows 2 years whereby imaging is done and Avantect is run, and we're trying to answer the question, how well does the blood test work, our Avantect test work in detecting cancer earlier? And if you combine the test with imaging, how well would it improve overall? So this is preliminary data because I like showing data. So here you can see across about 200 patients in the initial set of patients we've received, we found 1 confirmed. We predicted this individual, so this particular individual scores above our red line here, which is our test threshold. And it turns out this individual had confirmed cancer on their MRI. We didn't know that in advance. And then this other individual also is predicted to have cancer or an abnormality of their pancreas. And it turns out this individual has a germline mutation in this gene, and they have abnormal imaging on endoscopic ultrasound. So there's some follow-up there that we're suggesting. As we learn more about this, we'll obviously fill out this more. And then this last study with Kaiser is a very intriguing study because this shows you an example of how you can test patients and detect them earlier. So with this particular individual, they had 3 testing time points, T0, T1, T2. You can see at the beginning of the study, they had an MRI that showed a cyst with a solid component, but no definitive diagnosis of pancreatic cancer. And you can see that at that time point, that it's CA19-9 measurement, that glycan I told you about before that's used, but not always recommended. You can see that measurement was low. It was below the threshold. And so this was negative for cancer. At 6 months, you can see they became positive, but not sufficient until at 1 year, they actually had symptomatic disease. So they showed signs of pancreatic cancer. Intriguingly, across all 3 time points, the Avantect test was positive. So had this been done earlier, I think the outcome for this patient at stage 1 would have been even better, achieved much earlier. And so CA19-9 missed the pancreatic cancer diagnosis, and you can see that sometimes the imaging misses it too. So we're looking at more of these instances with this particular group. Our test is being used in these clinical trials. It's being used in a commercial context, but as a science team and thinking about Steve's vision, we're always trying to improve what we do. And what I'm about to show you is some early data that indicates in a future version of the test, we are going to be able to improve our sensitivity of detection and push it above 80% for early stage detection. Recall it was about 70%. We believe that this is trending towards 80% or even higher. And the way we do that is we combine additional features into our 5-hydroxymethylation signature. So the way we like to think about pancreatic cancer is that we'd like to change these statistics around that you see on this plot, right? Unless early stage individuals are very rarely detected currently, which is a very dire circumstance. Currently, with our test, we can capture individuals with a 68% sensitivity with a very high specificity of 97%. We can minimize false positives and capture individual pancreatic cancer. And I think coming later this year, the newer versions of our test that combines epigenomics with genomics, genetics, and glycan biomarkers is going to push us even further, and it's going to make for a very powerful test even further developed in the future that will enable us to detect individuals with disease. And so I'm going to stop there. I'm very pleased to have this opportunity to tell you about our pancreatic cancer test. We have other tests that look at ovarian cancer and multi-cancer. And if there's interest, obviously, I'm always happy to talk more about our testing strategies and our approaches. Everything that you've seen today is developed using the same 5-HMC technology. It's unchanged. By that, I mean the lab workflow uses the same approach irrespective of the test we're running. The part that changes is the machine learning and the algorithm that we develop to combine biomarkers further downstream. So it gives us, as a small team, small company running this, incredible power to really expand, further and release, these multiple tests in the short time that we've been able to generate and take the technology and bring the vision forward for early cancer detection. Thank you for all your attention today, and I'm always happy to take questions.
Oh, thank you so much, Sam. There's quite a few questions. This one is from a client of ours. So I am an HLI client. HLI has determined that I have a growing mass on my pancreas, about 2 centimeters, as I recall. So far, we are watching it. Will I be able to avail myself of this test before my next planned HLI visit in October? By the way, my dad died of pancreatic cancer. Is it okay to share this? It's okay to share this publicly. My answer is, definitely do the test. So, Sam, what do you think?
Yeah, no, I think so. And I think, Waywood, to your point, it sounds to me like you've done some very important imaging with the person who asked the question. Thank you for asking that question. And absolutely, that the test Avantect run on your blood sample in concert with imaging will really help your clinician figure out what to do. And yes, this is ready for your use, I think.
Uh, Dale, if you want to, I give you my longevity.com. I'll give you my email address and just send me an email. If you're interested, you can be our first client of the Avantech client. Actually, we are launching it, I think, May 1st.
Okay.
So,
Great.
yeah. And Keon has a comment, great talk and very exciting technology. I would expect for pancreatic cancer to result in type 1 diabetes from decreased insulin production, not type 2 diabetes from decreased insulin sensitivity. Do you know why type 2 diabetes risk rises with pancreatic cancer?
That's a great question. We don't know. We think there's a bi-directional relationship. By that I mean we think that the diabetes is actually a consequence of the cancer, and so and I'm not up on my um, I'm not on my up on my diabetes pathways, but one of the things that we've learned very early on is that apparently type 1 is very rare to occur with pancreatic cancer. So there's a difference in the kind of disease etiology with type 2 that makes it linked to the pancreatic cancer detection. So um, and it made sense to us that diabetes is frequently associated. 50% of individuals with pancreatic cancer have diabetes. 25% have new onset. So of that 50%, 25% have new onset. So there's a very strong association between the two.
Yeah, here's a follow-up question. Second, what counts as new onset type 2 diabetes? Most people slowly develop diabetes with a
Yeah.
gradually rising hemoglobin A1c progressing from normal to pre-diabetic to diabetic. Do we see a more rapid rise in A1C with pancreatic cancer?
Yeah, once again, excellent question. You do see that. So it's basically in this study that we're doing, we're focusing on a 6-month window between diabetes diagnosis and having the test run. There are groups, another group we worked with, I didn't talk about the study with NCI, they look at a 3-month window. So that time frame, to your point, long-standing diabetes only has a twofold higher risk of pancreatic cancer, not 6 to 8-fold. So yes, indeed, it is this sharp increase in hemoglobin A1c that leads you to essentially needing to test. And it's usually within 2 time points from hemoglobin A1c that you see this sharp increase.
Okay, here's a question from our first question from Dale. How does your broader test compare with GRAIL in terms of types of cancer detected and efficacy? I paid for GRAIL outside of HLI, and for the last 3 or 4 years, it has been negative.
Yeah. We frequently get asked that question. Our Avantect pancreatic cancer test is really tailored for pancreatic cancer detection. It doesn't, it minimizes the detection by virtue of the training of other cancers, and it specifically tries to facilitate this question about, is your risk higher and do you have the disease? So it's asking a different kind of clinical question. GRAIL's test is really focused in on, you're above 50, you're bound to get cancer sometime in the next 30 years at some rate. We'd like to capture you earlier. The other thing that we've looked at great depth is you can look within the GRAIL test and you ask the question, does GRAIL detect cancer better than us? And you find that they do not, because they're trying to optimize a test to look at many cancers. They cannot enable pancreatic cancer detection rates as high as the ones we've described.
No, yeah, I agree with you, Sam. Cancer is such a diverse disease. When you're trying to have a pan-cancer test, it's almost trying to build a car, which is trying to be a race car, a convertible, and
Right.
really comfortable Rolls Royce at the same time, which is almost impossible. So I'm personally, as a scientist working on early cancer detection for 30 years, so 40 years actually. I truly believe that we need to develop cancer-specific tests most of the time.
Yeah.
Actually, I have a question for you, Sam.
Yeah.
The you see different 5-hydroxyl methylation patterns in different cancer, right?
Yes, we do. And in fact, that's how we've developed our multi-cancer test. Um, the multi-cancer test has something that the pancreatic cancer test doesn't have. It has a separate algorithm that figures out which cancer is present. And that's because the 5-HMC signal is different in different tissue types and cell types. And so
Interesting.
we focus in on the ability to detect that by training the algorithm across multiple cancers. I think you also bring up an important point about cancer detection, and that is the way we've tried to focus has been on cancers for which there's no good detection modality. So pancreas and ovarian don't have tests. The CA19-9 is usually a test that's run once you have cancer to monitor how you respond to therapy. And so, and when we've developed our multi-cancer test, we try to focus on cancers for which there's no screening or early detection modality, because that gives you and gives the clinician more power to really offer a patient an aspect of their health that they otherwise wouldn't see, which is detection of certain diseases like liver, gastric, esophageal. These cancers don't have good detection modalities.
Yeah. Okay, so I have another question from Keon. Keon has a lot of questions. Third, is there currently a role for ordering Avantect in patients who are not diabetic but has high risk for pancreatic cancer, like polygenic with high polygenic risk score or suspicious lesion,
Yes.
like IPMN, or history of chronic pancreatitis.
Yes. So these are the risk factors that we believe are really important. Within, we know that we detect um, a certain number of pancreatic cysts, IPMNs, that have high-grade dysplasia, which means they're more likely to become pancreatic cancer. And when clinicians see those, they like to do surgery before they become cancer. And we detect those 88% of the time. And then certainly we detect pancreatitis at a high rate. In fact, that's one of the um, aspects of the tests that we remark on our patient reports. Not all pancreatitis becomes pancreatic cancer, so that's an important aspect. But any lesions seen on imaging or any known lesions to exist in the patient that essentially contribute to their risk become a reason to actually run Avantect.
Yep. No, that's great. So there's a question, will the Avon test be included automatically in the HLI blood testing panel? Our answer is this, not yet right now, because Sam is charging us too much money. Like all tests, at the very beginning, it's always expensive. I'm sure, Sam, you guys have probably invested hundreds of millions of dollars already into this test. And it's a very, very expensive R&D, investment in creating a new test. So we will launch it as a self-pay right now in our clinic, and we will, but for we do have members which paid, a concierge type of service. This might actually be included in the concierge service. Yeah. So the other question, is there a side effect or downside of doing the test outside of the cost?
Um, that's an interesting question. Not to my knowledge. I think that it's certainly true that when risk is increased, there's
Uh,
definitely an upside to doing the test. So in any
uh,
of the areas of risk that I described, familial history, new onset type 2 diabetes, pancreatic cysts, there's definitely a benefit to doing the test. And I would say that you could judge the downside as being, gosh, what would be my chance of being detected as a false positive?
uh,
And we deliberately made our false positive rate as low as possible. So again, no test is perfect, right? Grail's test has false positives and so on. We had the same rate of false positives, which is extremely low, like 2%, I think I showed you.
yeah. Well, this is the elusive goal in cancer detection. We want ultimately a system can provide 100% specificity and 100% sensitivity. Okay, so in cancer biology, we have not achieved that yet. For monogenic genetic disease like cystic fibrosis and sickle cell anemia is actually pretty close to 100% sensitivity and 100% specificity. So what we are doing at Human Longevity is by combining whole genome sequencing, which can identify those 10% of the people who has high genetic risk factor, like Angelina Jolie has a BRCA1 mutation. We think actually, the Black Panther actor, probably has a Lynch syndrome. They're born with a high genetic risk factor.
Right.
Today, you can identify those patients, but then you have imaging. So, and then you have liquid biopsy. I think there's also another layer, which is proteomics. We are working with Tony Weiss-Corey at Stanford to look at 7,000 proteins. So when you combine all that together and do it longitudinally, so don't just do it once in 20 years, but if you do it, once a year, even some of these protein marker like CA125, if you did it once a year, you can see the trend of these protein markers. I think we're getting closer to the elusive goal of 100% sensitivity, 100% specificity. And
Yeah.
that's what Human Longevity is trying to, do it inside a clinic. So this clinic actually can provide all these tools, available to our clients, because these tools are complex. Because we hear people do piecemeal. They go to do Grail, they go to Mayo Clinic for something. They go, but it's actually the art of putting them together is actually very complex.
Yeah.
It's almost like a Michelin star restaurant. You can't just buy all the raw material and have your own Michelin star restaurant. It's almost like putting the restaurant together is actually a complex process. We have been doing it for 12 years now, and we're improving it, every year. And with the new tests like Avantect, we are improving on pancreatic cancer testing and ovarian cancer testing, which is so needed, right? So
Exactly.
there's
Absolutely.
a comment from Madison. This is exciting for our clients that have IPMNs, which we follow closely with imaging. But a test like this can provide another layer of screening. Yeah, yeah. So
Absolutely. I think that's
yeah,
exactly right. Exactly
yeah.
right.
So Madison, I was at the Mass General last week talking with Dr. Keith Lillimore, the chief of surgery at Mass General. He's one of the best pancreatic surgeon in the world. He is the one saying if we have a liquid biopsy like uh, Avantect, he is willing to do a trial at Mass General because they do see a lot of IPMN. It's like a polyp in the pancreas. But some polyp will never grow into tumor, but some polyp will grow into a tumor and kill you eventually. So now with a mass, 5-hydroxyl methylation test, I think we are actually truly creating a new tool to kind of separate people from a polyp never will kill you, and a polyp might actually kill you in 3, 4 years. I think this is really huge. And another question, do we have an idea that what may be causing some of the false positive?
Yeah. So that's a great question. We in a separate study we've done, we have noticed that sometimes any form of immune modulation, whether it's through medications, whether it's through vaccination, can cause a false positive. In the Kaiser study we did, there was a strong association of the individuals within a month of their COVID vaccine. Essentially, that triggers the immune system. Some of the cancer signals that we're detecting from the DNA floating around in blood essentially capture the immune system, and
Wow.
they also are cancer signals, and so that can cause a false positive. So, that we're still trying to learn the full spectrum of these, but they tend to be rare, as you've seen. We've tried to minimize the signal from those false detection events.
So what is the positive predict value right now?
Yeah, so if you look in the kind of high risk populations,
Yeah.
it's about 20%.
Okay. Okay.
Yeah. And the NPV, the negative predictive value, is about 99.6%.
Yeah. That's so basically, if you test negative, you can sleep well.
Right.
If you test positive, it's 1 out of 5 people basically
Correct.
is positive.
Correct.
And
Correct.
so the other 4 people basically need to go through the other imaging and all that to rule
Correct.
out cancer. Yeah. So here's a question. If someone has pancreatic cyst on images that are just being followed, would it make sense to do your test? Yeah, my answer is yes.
Yeah. Yeah.
Uh,
Correct.
yeah. There's a question say, do we routine get this test at HLL? No, we're actually just launching this brand new test in May. So HLI is officially launching this pancreatic test in May. What what does the test cost? Wait for a few days. I think we're still trying to figure out the cost. Okay, so
Yeah,
uh,
that's a question I'm not allowed to answer because I'm a
yeah,
science guy.
okay, so
I can't tell you what the test costs. Some of my colleagues, Daniel Scalfo, and then a few
yeah.
other folks online will be better poised. But yes, we should follow up on that one.
Yeah, we will follow up. We will publish the pricing
Yeah,
probably in a week or two. But my experience of all tests eventually gets cheaper and cheaper. So when actually human longevity was doing whole genome sequencing, I believe our cost at the time was $10,000. But today the cost is coming down to about 15, $18,000, a clear cap whole genome sequencing cost. So, but when Sam, you were involved in decoding the first human genome at Celera. At the time, I think Celera cost a little bit over $100 million, right?
that's right.
But the US government spent $3 billion to decode the first human genome. So that's only 25 years ago. So 25 years ago, to decode a human genome cost $3 billion. Today is around about $1,000, $2,000. So
Yeah.
all these costs eventually will come down in price. So we are obviously the innovators, and but I think our client base is willing to pay a little bit more to be the first group of people don't die from pancreatic cancer. I think this test for Steve Jobs will be worth $1 billion. Uh,
I think for the most part, just the idea of you have to trade off the cost of the test in relation to health economics. And
yeah.
we did do a study that showed, the health economic benefit of running our test at our current price, whatever that is, has great value because you're saving patient lives, right? The cost on an annual basis can be realized. And so that's you don't have to worry about, it's a hand waving reason that the cost is good, that we've published something in conference proceedings that show that. So happy to share that information with you.
Okay, so again, there's a question about when the test will be available. You probably have to wait for a week or two for us to have the commercial strategy for this test, but we're going to make this test available in May. Uh, there's a question called, what is the LOD95 of tumor fraction for this test? I don't even understand this question.
Yeah. So no, I do.
Okay, that's
Yeah.
great. Okay, so
So we don't in the world of minimal residual disease, the LOD is used, and tumor fractions measured. Intriguingly enough, I didn't include the data on this, but um, we've been able to compare our test score and how well we detect in relation to tumor fraction. And we're getting down to 1% or lower tumor fraction measurements, whereby we can test a pancreatic cancer positive. So if you think about it, cell-free DNA, the DNA is floating around in your blood, only a fraction of that, as you know, I think, given you've asked the question, it comes from tumor cells. The rest of it comes from all the surrounding tissue, immune cells, other white blood cells, other tissues, and the supporting tumor microenvironment. What we're detecting is a change in all of that due to cancer. And so some of that's tumor-based, but some of it's not. And so we actually find when you look across our multi-cancer test, we can detect down to quite low tumor fractions. And this is not just a pancreas specific result, this is a pan-cancer result. So very pleasing to measure that 5-HMC signal in that context.
Yeah. So maybe we'll answer Eric. I missed my favorite doctor's question. Thank you, Dr. Levy, for sharing this groundbreaking work with us. Is there an advantage to taking the Avon test for pancreatic cancer only, or is the multi-cancer test equally as effective? If both tests are equally effective, why wouldn't we always take the multi-cancer test? Thank you.
Yeah, I think that the ability to, again, it comes back to the ability to detect a single cancer at a higher level versus being able to detect multiple cancers at the highest level possible. It's hard to optimize as well multiple cancers as it is single cancers. So we've found we're in the early days of comparing our data for patients with pancreatic cancer using our pancreatic cancer test versus our multi-cancer test. Our performance metrics for our multi-cancer test
Well,
appear to
somehow we
be very
lost your
similar.
voice, Sam.
Oh, I'm
Can you
sorry.
hear me?
Yes, I can hear you. Yes.
Or is it just on me?
Can you, I I'm not on mute as far as I can tell. Can you hear me okay?
Yeah, no, I can hear you now.
Okay.
Maybe my internet. Yeah.
Yeah. So I think that so again, the point is the optimization is better when you do a single cancer test. And when we've looked at our performance of our multi-cancer test for pancreatic cancer, it's very similar to our single cancer test currently. However, if you recall at the end of the presentation, I gave you a foretaste for the future, and there we think we can improve our test performance. And I think that will only be a possibility to do in a single cancer mode.
Yeah. There's a question, do you know any causes or correlationships with pancreatic cancer?
Could you ask the question
Or,
again? Any
yeah,
causes?
do you know any causes or correlations with pancreatic cancer?
The causes for pancreatic cancer tend
Yeah.
to, yeah. Well, I think that, I mean, there are a lot of risk factors that are actually common to many diseases, right? So smoking is one. There are other risk factors around obesity and so on. I think, but that's not specific for pancreatic cancer. That's specific to other diseases. Um, but I think that, or common to other diseases. But I do think that for for the areas that I covered whereby risk is higher is, a genetic component like family history, and it's clear that there's a genetic component that is present for pancreatic cancer. If you have gene mutations in your germline genome that put you at higher risk, there's a cause. That's a cause for disease ultimately in your lifetime. And then the other piece is the the effect of diabetes, right? So the fact that there's a sporadic cancer, it's unclear again whether we think it's the cancer causing the diabetes, but that's still not 100% certain. So that there is maybe some relationship there. So those are the areas that I'm aware of. There may be others as time goes by that we'll actually learn. put patients at higher risk.
Yeah, based on human longevity's 10,000 clients and a 10-year worth of follow-up, we really think people who has familial history of cancer should pay a lot more attention. And also, obviously, people has high risk genetic, polygenic risk score, which we update our score every year. There's a new publication in New England Journal of Medicine just just a few days ago on prostate cancer. They actually have more, you know, supposedly more accurate polygenic risk score for prostate cancer. We're upgrading our client's genome on this new algorithm just published a few days ago. So it's an ongoing improvement process. And people has BRCA2 mutation or BRCA1 mutation definitely needs to be uh, extremely careful with pancreatic cancer. Uh,
Yeah, that's
so
right.
yeah, there is so what is a false negative rate for Avantect test? That's 0.4%, right?
Yeah, so that's right. It's about zero point 0.4, right? So fraction or 40% false negative rate.
Yeah, because the
False
negative
positive rate. I don't know if the question was around false
the
positive
false
is
negative,
3%. Yeah. Okay, great. Very good. Yeah.
right? The false negative rate or the negative predictive value is 99.6%.
Right.
So the false negative rate is 0.4%. It's less
Yeah.
than 1%. Yeah.
Correct.
So this is the amazing test. Yeah. Yeah. So if I test negative, I'll feel good. Okay. So yeah.
Correct.
Yeah. So, okay, there's one comment. This is such an exciting news. Most of our patients are very concerned about this type of cancer. So it is great to be able to afford more screening tests. Okay, that's a comment.
Yeah. Okay.
Okay, will any insurance plan pay for this test? For example, say someone insured by Kaiser can have tests taken at their local Kaiser branch?
That's a little bit why we're doing these studies to work with these different groups to show the value of the test in their clinical context. And with our colleagues who work on reimbursement, Danielle Scalfo, who I mentioned, we're trying to work towards the clinical studies needed to convince payers to reimburse, whether it's Medicare, whether it's private payers, groups like Kaiser, and so on. So it's on our strategy. And definitely we're driving towards that. Stay
Yeah.
tuned.
Okay.
And I'm just told by my colleague Danielle that indeed some payers are reimbursing already, and we can get you that list.
Okay. Okay. Yeah, probably go to our website, and we will provide that list, which
Yeah.
insurance is starting to pay for these tests. And the last question is, you mentioned there will be a type 2 test, which will be available in Q3. So the question is, are you sure it will be launched in Q3?
Yeah, of course. I think
Okay.
one thing that we're trying to do is the Q3 availability of that test is really to enable certain clinical studies that I haven't mentioned to complete in time, and they will essentially prove the value of that test. So it's a little bit a combination of the test readiness associated with that clinical study. So yes, stay tuned. I think that the benefit of doing the test now is as high as waiting for Q3, if that was perhaps the question, because that difference and that change in performance is not significant for anybody running the test now. It will be significant for somebody who's never run the test, obviously, and so we would encourage, if the test feels right for you, if it's appropriate, your clinician and working with HLI feels it's appropriate, there's no timeline right now.
Yeah, yeah. No, this is such an important, innovation. I remember, the first company, I joined Dr. Craig Venter's first company, Human Genome Sciences. Our CFO, uh, I was talking to him in October, in Christmas time, he's already dead. He didn't actually in October, he didn't even know he had
Yeah.
pancreatic cancer.
Yeah.
But I think end of October, he was diagnosed with pancreatic cancer and he died in December. So late stage pancreatic cancer does cure very quickly. So, so this is really the, it's a, uh, if people has familiar history and high genetic risk factor, you know, personally, I think it's a no-brainer to, do a test. It's
That's right.
it's negative predictive value is 99.6%. If I test negative, I'll be so relieved, So now, Sam, thank you so much. This is so wonderful. We are actually also, really looking forward to the ovarian test launch. We're right now focusing on the pancreatic cancer
Right.
launch.
Right.
But the ovarian cancer really has no solution. I used to work with Dr. Robert Bast. He's the one discovered
Yep.
CA125 like
Yep.
30 years ago. And we haven't had anything new in ovarian cancer for a long, long time. But I think, I'm really looking forward to the clear node test for ovarian cancer. Yeah.
Yes, and Wei, thanks again. The ovarian cancer test is out there. We can talk
Yeah.
about it on a different time. Always happy to do that. And
Yeah.
thank you for all the great questions and everybody's attention today. We really appreciate it. Don't
Yeah.
hesitate to reach out to us.
Yeah, don't hesitate to reach out to me on LinkedIn. So it's really fascinating. Time to have the greatest solution to prevent cancer. Yeah,
Great. Thank you
yeah.
again.
Okay, thank
See
you.
you.
Okay,
Cheers.
bye bye.
Bye-bye.
Okay. Thank you, Paul.